Right. Okay, what am I doing? Right. What am I doing? Hi, Andres. Sorry, I am literally. Hi, Daryl. Hi. I just spotted you on the other one there. How are you? Find link. I'm actually on. Hi. Sorry, guys. I'm running so late tonight. Today's just been one of those crazy days. I do not know what is going on with me today. Daryl, how are you? It's so nice to see a face. Well, see a profile of an ex-student. How are you? How are things? How's uni? Guys, if you want to come and join me, I am on my YouTube channel now going live, and the link is there for the Zoom lesson. Exceedingly well, better than this. Oh, that's great. That's really good to hear. I'm really pleased to hear that for you. It's crazy to think that this year's year 13 are going to be doing their exams in like literally 7 weeks. Oh no, that's not good. Do you know what happened? The notification clicked on it, but my internet ain't great. Oh wow, you're in Romania. How come? Sorry guys, just chatting away to one of my ex-students here. Daryl, who is studying oh, don't be embarrassed. We all do it. I have days when I cry. Even teachers have bad days. Like that's the thing for you to note. Gotta love a group poster. Well, that does tend to happen in most group sessions. There's always one who leads. Hey, there's nothing wrong with getting frustrated and crying. That's one of my go-to. My frustration always comes out in tears, and it looks like I am sad, but I'm not. I'm just angry and frustrated, and I can't control it. These things happen, and you know what? You live and you learn. I've done it in meetings as well. Like it's so, I get it, it's embarrassing, but once you get past it and once you realize, you know what, you just don't need to worry about these other people. If they're not putting in the effort like you are, they're not worth your time of worrying about it. Yeah, I have done actually. Yeah, it's a bit crazy, but yeah. It's not great, but there we go. Right, so I am over on my YouTube channel and I am going to actually go into some papers. So if you are joining me tonight and you want me to cover specific topic areas, then please do ask away. If not, then I will just pick at random a paper and start doing some questions based on that. So, oh, I've just noticed there's a whole load of people that I haven't let in. Sorry, guys. There we go. I'll bring you in here. So those of you who are joining me in the Zoom live, if there is anything you want me to do, then please ask away. Otherwise, I will just pick a random paper for us to cover in the session today. Try to think what we've not done in a while. Not really done a paper one for a while, have we? Okay, from the source cell to the combining cell and then into the sieve tube element. This decreases the water potential of the sieve tube element. So naturally water moves from the xylem to into the osmosis. And then what happens to the water? What does it move back? Right, okay, so you want to look at a bit of translocation. Let me do that then. Let's start with some translocation because you've asked the question, and that's the whole point in these sessions. So if you're talking about translocation, you've got it absolutely right in the fact that by loading the sucrose, yes, Lynn, I will cover that as well. By loading the sucrose into the phloem sieve tube element, what you're going to do is you're going to affect that water potential. And if you're affecting the water potential, water will move by osmosis into the phloem. I'm trying to move this. Where are you going? Move down here. So it's going to move into the phloem. And so when it moves into the phloem, what you're going to get is, this is on a change in the water potential. So, oh no, I'm the wrong way. There we go. Right. So here we have it, that we have got the source where we're going to produce the sucrose. So let me just, I'm just moving people around right now. So we've got the source. The source is going to be doing photosynthesis. This is going to be where you've got photosynthetic pigments. So they're going to be photosynthesizing, light-dependent reaction, light-independent reaction. We're therefore going to have our triose phosphate. The triose phosphate is then going to be able to be converted into glucose. Glucose and fructose being added together gives us sucrose. So this is all linking it together. Then, yes, so this is a bit of photosynthesis, which is module 5. Then it's going to be looking at module 3, which is your transport and plant. So it all interlinks. Module 2, osmosis. So you need to think of biology as the bigger picture now, and not just as this is module 3, this is module 5, this is module 6, because that's not what they're going to do in the papers. Paper 1 will ask you questions on module 1, 2, 3, and 5 interlinked together. So photosynthesis is module 5, translocation, which is what we're going to look at now, is module 3, but they're all interlinked. So you need to, as I say, see that bigger picture. So we've got our photosynthetic pigments absorbing the light. We've got the light-independent reaction, light-dependent reaction, making the triose phosphate. That can then be made into glucose. Glucose and fructose are biological molecules. Glycosidic bonds, we've got our disaccharide. So we've got our disaccharide, and that disaccharide is going to be loaded into the phloem. So you know that your water is going to move up through the xylem vessel because of the cohesive and adhesive properties. So what you then need to understand is by loading sucrose into the phloem sieve tube element, that is going to lower the water potential. Water will then move from surrounding cells as well as the xylem. So Andreas, what you asked about it and mentioned about it going from the xylem. Don't forget it also moves from the cells surrounding. Any cell which has a higher water potential than that of the phloem is going to lose water by osmosis, because remember, osmosis is that net movement of water from a high water potential to a lower water potential. So water is going to move into the phloem. And then what you have to remember is, if I have a bottle of water and I try and squeeze it, if there's no air and it's just filled with water, if I try and squeeze it, what's going to happen is it's not going to be able to be squeezed because of the fact that it has this rigidity and incompressibility. So what's going to happen now is, because of the incompressibility of water, we're going to build up this hydrostatic pressure. So that hydrostatic pressure is now getting higher and higher and higher. So what's going to happen is water is going to move from where there's a high hydrostatic pressure to where there's a lower hydrostatic pressure. So that's going to result in the water flowing to an area which is lower. Now, I've shown it moving down, but remember, it can move up as well. So it's going to move in either direction depending on where there is a lower hydrostatic pressure. So when you get to, let me just add in, hold on, this and this, and you get to the unloading, what you've got is wherever we're at the sink, we're going to unload the sucrose. So therefore, diffusion through the plasmodesmata is going to then allow from it going from a high concentration to a lower concentration. And then what you have is that sucrose can either be converted to glucose, respiration, or it could be converted into starch, storage, or it could be converted into amino acids for growth, or it could actually be converted into lipids. Whatever needs to happen with that source, so the sucrose that was made at the source is now going to the sink and is now going to be utilized. What you're then going to have is that they are then going to result in the sucrose, because it's been lost, has now increased the water potential of the phloem. Because we're now increasing the water potential of the phloem, that's going to result in water leaving to surrounding cells. So that sucrose leaving, the water then leaving, is what's keeping it at a lower hydrostatic pressure so that that movement up and down still occurs. Has that answered your question? Andreas, does that help? You are very welcome. Okay, so Lynn, you've asked for some questions to do with genes. Are you specifically talking about the regulation of genes, or are you just talking about the different versions, so alleles and the inheritance part of it? Which bit are you actually wanting to look at when it comes to genes? Alleles and inheritance. Okay. Not a problem. Let's go for this then. Okay, so just as the last bit to finish off, because I have this diagram here. As I said before, remember your sinks can be down and above because that lower hydrostatic pressure could be below or could be above. So your source could be whatever that photosynthetic pigment is, i.e. the photosynthesizing tissue, leaves and stems, or it might actually be a storage organ. So it could be a seed. So if you were looking at, for example, germination, or it could be a taproot or a tuber. Lily, if you want to jump across, I am on my YouTube channel and there is a link for the actual Zoom lesson if you want to. I've had characterized the facts. So Lydia, again, same to you. I am on my actual YouTube channel if you want to jump across and come and join the live that I'm doing now. I'm streaming it there and also doing a Zoom lesson. With regards to the content, it is difficult if you don't have the knowledge of the content, but it's about making sure you've got the keywords, making sure you've got the foundation parts that you can then embellish on whenever they give you a question. So Lynn, let's start with some alleles and inheritance for you then. So if we're looking at population and we know about variation, so you should know about variation, you should know about whether it's intra-specific or whether it's inter-specific variation, intra within the species, inter between the species. You should also know that when we talk about variation, we normally talk about it when we look at the phenotype. So the observable characteristics that you're going to see within a individual, whether that's a human, whether that's an animal, whatever that is, it could even be a plant. What are the observable characteristics that we're seeing? Hair color, eye color, petal color, seed shape, whatever that may be, that is down to the genetic information that they possess. So then you need to understand, well, okay, that genetic material, which is found inside the nucleus or in the nucleoid, if we're talking about a prokaryotic cell, that genetic material can then be influenced also by the environment for which the organism lives. So if I right now go outside during the daytime, it's glorious, it's lovely, it's sunny. That sunshine is going to potentially result in my phenotype being different because I may get a tan. But after a certain amount of time, that tan will disappear on me. So what you have to understand is the environment can have an effect on the observable characteristics, but the genetic makeup that you have is set. It's inside all of your cells. It's inside the nucleus. So What you then have to understand is that an allele is two or more alternative forms of a gene. So here I'm going to look at it starting with our plants. So we have our seed color and we have our petal color. So I have my chromosomes, and my chromosomes, just as a reminder for those of you who are year 12, your chromosomes are linear if we are talking about eukaryotic organisms. I'm talking about plants here, so I know that they are eukaryotic. Classification, module 4. I know that the chromosomes are linear, and I know that those chromosomes have DNA, which is associated with histone proteins. So, Aya, when you said earlier that you wanted to discuss, sorry guys, one second. Okay. Sorry about that. I was just reading a message about sausages. You wanted to discuss about which module were we doing. What I'm trying to show you here is that we're looking at our different modules all coming together. So what you have is DNA associated with histone proteins, linear chromosomes, eukaryotic organism, because I'm talking about a plant. That will be some little hints in the question. If they told you seed color or petal color, they expect you to be able to put those two pieces together, and they may then start asking about questions to do with nucleotides and nucleic acids. Okay, so what do we need to know then? A dominant allele. If we have a dominant allele means that that gene will always be expressed. Now, what that means is that they're actively influencing the organism's characteristics. So if you have a dominant allele present and you only need one, that whatever that characteristic is, that will be expressed, that will be present, that's what we will see in the phenotype. So I have got brown eyes, so therefore I know that my brown eye gene potentially could be dominant to the blue eye gene. Well, it is dominant to the blue eye gene. We know that. We teach that at GCSE. So because my brown eye gene is dominant to the blue eye gene, I have brown eyes. My partner has blue eyes. My daughters both have brown eyes. Well, they could only inherit one allele from their father, one allele from me. Therefore, the two that they're going to get is a blue and a brown allele for their eye color. Well, the fact that they've got brown eyes means that brown is dominant because that's the one that is going to influence the organism's characteristic. That's what you see. Now, to be able to see the recessive allele, the corresponding small letter, they must have two of those alleles present. So that's really important for you to understand. Anytime they tell you that there is a recessive allele, if that recessive trait is showing, whatever that may be, in this case, yellow seed color and purple petal color, if that is the recessive trait, the only way we will see those phenotypes is if they have two of the recessive alleles present. So that's really important. So then we need to understand, well, what is a gene? A gene is obviously a section of the DNA. So it's a section of that DNA sequence. So a gene mutation, whatever that may be, whether that's an insertion, whether that's a deletion, whether that's a substitution, a gene mutation is going to potentially, and I'll say this potentially, cause a permanent change in the genetic composition of an individual, causing a variation, unless that mutation was silent. If you have a silent mutation, then it's not going to do anything. Even though there's a permanent change in the genetic composition, the phenotype might not actually show any differences. Oh, thank you, 27, and thank you that you're watching the YouTube. I'm actually on YouTube just now. If you join me there, you'll be able to join my actual live session where I show the screen of what I'm talking about. Okay, so back to this then. We have this gene mutation. We've got the composition. We've got individual, this causing of a variation. So then you need to understand about whether we have got what's known as homozygous allele. So if we have homozygous, we're saying that those alleles are the same inside the zygote. So homozygous alleles in the zygote, and for those of you who don't know, a zygote, excuse me, is a fertilized egg cell. So when you have a fertilized egg cell, a zygote, what you can have is you can either end up with homozygous alleles for a specific gene, which therefore means that you're going to have, in this case, either two dominant alleles, capital G, or two lowercase g's, which means yellow, capital P, pink, lowercase p, purple. So the genotype is the combination of alleles, capital G, small g, capital P, small p. That's the genotype. The phenotype, the observable characteristic, was the seed green or was the seed yellow? Was the petal pink or was the petal purple? So that's the phenotype, the genotype, that combination. And if it's the same, we know that they were homozygous. So this is obviously us looking at a single gene. So we're looking at a monohybrid cross. I will go into dihybrids as well. Heterozygous, if the alleles are different, they are therefore called heterozygous. Hetero, different alleles within the zygote. So in this case, we would have capital G, small g. So we can see the capital G because we've got the green allele gene. We've got the yellow allele gene. Therefore, in my pair, because remember my chromosomes are homologous chromosomes, which means that they have the same chromosomes. No, they don't. Let's try that again. They have the same genes in the same locus, which means position. Therefore, they are the same length. So we know if we're looking at humans that we have 23 pairs of chromosomes from chromosome number 1, which is the longest one, all the way down to chromosome 22. They go from the longest right the way down to the shortest. Then we have our 23rd pair, which is either the XX or the XY. They are different lengths, especially if they are the X and the Y chromosome. Right. So now let's look at inheritance and look at the monohybrid crosses, and then we'll look at dihybrid crosses. And if you have questions, by the way, as I'm going, please do ask the questions because I'll just keep rambling. Are you meant to be saying what you're writing? No, unless, Tom, you jump over to my YouTube channel or come join the live Zoom that I'm doing right now. So I'm actually doing it live streaming on my YouTube channel, but this obviously is for those of you who come here on my TikToks. I've moved across now. Okay, so over to this then. If we're doing a monohybrid cross, I'm going to use Gregor Mendel, and look at the inheritance of different characteristics in the pea plant. That's the common one that we always use. They're not going to make it simple like that. They're not going to give you Mendel's pea plants unless it's a low-level question. If it's a low-level question, they will give it to you, but that will be only a couple of marks available. But if I have a monohybrid cross, I'm going to look at one gene only. So I'm going to look at whether they've got tall or short phenotypes in the pea plant. And it was all before he knew anything to do with chromosomes. So what you can see is if I have a tall phenotype, the genotype, if it's a monohybrid cross and they are homozygous, this is dominant, therefore homozygous dominant, TT, capital T, capital T. If it's short, short is recessive because I know capital T gives me tall. I therefore need to write the corresponding lowercase. Then we have what would be in the gamete. So someone asked me on here about meiosis. So if you think about meiosis and you think about the fact that it's a reduction division which is going to result in a diploid. So this is what we call a diploid cell because it has two of the chromosomes. Lynn, I'm going to come to that in a second. I'll come to when it would be a capital T, small t, because you can see that they all produce that down here. It's a diploid cell when we're looking at the somatic cells, when we're looking at all our body cells. The only time we're looking at the haploid cells is when we're looking at the gametes. So the gametes are haploid. So if we're talking about a plant, we're talking about the ova or we're talking about the pollen. So the male and female gametes that are produced through the process of meiosis is going to reduce the number of chromosomes. We're going to go from a diploid cell, which has two sets of every chromosome, to a haploid cell, which has one set of every chromosome. So therefore, if I am going to cross a homozygous dominant with a homozygous recessive, all of the offspring will always be heterozygous because they can only take one from one of the parents and one from the other parent. So therefore, if there's a homozygous dominant and a homozygous recessive and we mate them, we will always get 100%, which is heterozygous. So now we take this F1 generation, and what we're going to do is we're going to put that and we're going to cross them together. So what we're saying is we're going to take a TT and cross it with another TT. So we've got two heterozygous that we're going to cross together. So if I put them into my genetic diagram, I put one of the potential alleles, the capital T, and one of the other alleles. So I've got a capital T here. I've got lowercase because that was what was in the parent genotype, and therefore that's what's going to be in either of the gametes. They're either going to be a capital T or a lowercase t, and the capital T, lowercase t, bring them across and bring them down. You get tall, you get tall, you get tall, you get short. So therefore, I have got these three phenotypic ratio, three which are going to be tall observable features, and one which is going to be a short. Now this is somewhere, so the phenotypic ratio is a 3-to-1 ratio. If they ever ask this, if they ever ask you about a genotypic ratio, wow, I cannot speak tonight. If they ever ask you about a genotypic ratio, you should therefore have capital T, capital T. You then have capital T, lowercase t, two of them, and then lowercase, lowercase. So therefore, if I was looking at this ratio, I would therefore have one capital T, two capital T, lowercase t, and one lowercase t, lowercase t. So when you get to the Hardy-Weinberg equation, that's where this comes in. So the T, capital T is the dominant, so it's most likely the offspring will inherit the allele for tall. So only if the gamete that carries the capital T is the one that is actually going to be fertilized. So to answer that question, it's only the ones which are the capital. There is more likely a chance of inheriting a 3-to-1 ratio. Now, remember, when we do this, whenever we look at genetics and we look at the possibility of it, we're expecting a 3-to-1 ratio. So if I had, I'm just going to make this number up, 1,400 plants that I observed, how many of them should be tall would be, I would need to divide that number by 4. I can't do it because my calculator's up there. So used to get my phone out. So if I get my calculator and I do my 1400 divided by 4, and then I times, that would be 350. I would expect 350 of them to be short, and then I would times that by 3. No, still, it's clear. That times that by 3. So therefore, I would expect 1,050 to be tall. So if they were potentially going to ask you about a chi-square test, statistical test, this is how they could do it, in the fact that they could say, what would your expected be? Well, I know if I have 2 heterozygous being crossed, my expectation is a 3-to-1 ratio. So therefore, I would expect a 1,050 of my plants to show the tall phenotype and only 350 to show the short phenotype. Does that make sense? Right now, for those of you who are joining me on here, I am covering the genetic side of inheritance, patterns of inheritance, module 6. Did you do SQA biology? Yes, I did, but I only did my what would it be? It was the higher. I didn't do the advanced higher, and we're going back a number of years before that. Good. Dihybrid crosses are a bit scary, but not if you really know what you're doing. But just to be clear, guys, okay, when I go through this and talk to you, I hated genetics. Even at university, I hated genetics because I really struggled. It is a really tough topic to get your head around. It's not easy, but the more you do it, the more you practice, the easier it will become. It's about repetition and knowing the key bits about whether it's dominant or whether it's recessive. But it's hard. It is hard. And anyone says it's not, they may find something else hard. Different people have different strengths and weaknesses. Okay, sticking with the monohybrids for a minute, and then I'll go to the dihybrids. So this time, what I'm going to do is I'm going to do a monohybrid cross, but this time I'm going to do a test cross. What that means is I'm going to take a individual, which is tall. I don't know whether it is homozygous dominant or whether it's heterozygous. I can work this out by doing a test cross, by crossing it with a known genotype, a homozygous recessive. So if I take a tall and I take a short plant, now remember that tall plant could have been capital T, but if it were capital T, and I crossed it with a homozygous recessive, all of my offspring would be heterozygous. I would get 100% of them, which are going to be tall heterozygous plants. So therefore, if I don't get that, if I get a different combination, so if I get a two shorts, a 1-to-1 ratio, 2 tall, 2 shorts, that would therefore mean that I had to have had a heterozygous individual. So that's why we call it a test cross, because we're testing whether or not that tall individual was homozygous dominant or whether it was heterozygous, because we are testing it with a homozygous recessive. Thank you for the rose, Angelo. Sorry, I'm trying to do both screens at the same time. So if we look at this, then you can see that only these two would end up with a tall phenotype because they have a genotype which is 1 dominant and 1 recessive. They're therefore heterozygous, whereas these two are homozygous recessive. So again, if I use my numbers 1,400, I would therefore expect there to be 700 to 700 of them, tall to short, roughly about that number, if I am looking at the expected ratio. So if we then look at the dihybrid cross, and we now bring it in with a bit more. So now I'm going to bring in a dihybrid cross. So what I'm going to do this time is I'm going to look at two different traits, yellow and round seeds, and I'm going to look at the genotype. So what can you tell me about the genotype for each of these individuals? So those of you who are in the session, what would you say? Is there anything you can tell me about these individuals? Good, Andres. They're all heterozygous. How do you know? Well, you can see that they all have at least one capital and one lowercase. So therefore, what I have done is I have taken two heterozygous. So they could give you in the exam the information in the fact they've shown you the genotype, and they then expect you to tell them that that's heterozygous. So what if I had a capital Y, capital R, small r? What would you say then for this individual? What's dominant, Lynn? Tom, the answer to that one is make sure you know your content and then look for the hints in the question. When it comes to AO2, when it's looking for you to apply that knowledge, that's what I'd say. But if you're interested, I am actually on my YouTube channel live streaming right now where you can actually join the live session that I'm doing. They are homozygous for the Y. Be more specific, Andreas, on that one. Lynn, you're saying dominant, the Y and the R. So they definitely have a dominant allele. You're absolutely right. Good, Andres, you're saying that they're heterozygous for R. That is true. So therefore, we can see this gene, they are heterozygous. But for this gene, they are homozygous. But try and make sure when you are giving the information, you be specific. They are homozygous, dominant. So you said they were homozygous for the capital Y gene. When it comes to you answering it in the actual exam, say that they're homozygous with the dominant allele. So it's a homozygous dominant or a homozygous recessive is what we would tend to use. Okay, so in this example, we know they're heterozygous. They're both heterozygous. So if I am going to look at meiosis, how do they separate? How did they line up at the equator? And how did they then end up being segregated? That is all through the process of meiosis. Meiosis 1 is about the homologous chromosomes being separated. Meiosis 2 is about the sister chromatids being pulled apart. So try, whenever you're revising this, try and get those keywords in there. Try and give the information that the examiners want to see, okay? So what have we got then? Well, we can see that we've got a capital Y, capital R. We've got a capital Y, lowercase r, lowercase y, capital R, lowercase y, lowercase r. So there is 4 potential combinations that you can have when we're looking at a dihybrid cross. So then you literally are just going to put them into your Punnett diagram and then fill the grid out. That type of question would be a 5 marker, and it is just about taking your time to draw down and across. So bring it down, bring it across. My hint to you would be wherever you see a capital, so if I use this one, always write the capital first. So in this case, is it one capital R, lowercase r, write the capital first so you know that dominant is there. If you were to write this capital Y, lowercase r, capital R, you're still correct. You won't be penalized on that one. However, just make sure that when you do that, that you remind yourself that that capital R is present, and therefore that would give a round seed. So if you're going to go through and you've got your generation, your F2 generation all filled out, and I've used color coordination, you should then be able to work out your phenotypic ratio. So when we're looking at the phenotypic ratio of a dihybrid cross, now this is one you should know. You should be able to pull this straight from your head that the dihybrid cross is a 9:3 to 3:1 ratio. However, okay, this is my warning to you all. Students, memorize. You can memorize that, absolutely. A 9:3 to 3:1 ratio. If I have a hetero with a hetero, and I am doing a dihybrid cross, I know my expected ratio is going to be a 9:3 to 3:1 ratio. I know that. But if you memorize that and then they throw a curveball at you and they give it to you where it's not heterozygous with heterozygous, for example, they give you a homozygous dominant for one allele and a homozygous recessive for another, it completely changes your ratio. So just be very careful that you're taking the time to read what they've given you. Okay, so if we go back here, if I do a test cross, remember my test cross is that I'm going to use my recessive, if I can spell recessive, and it's going to be homozygous. So I've got a recessive homozygous. If I do a test cross with a dihybrid heterozygous, my ratio this time is a 1:1:1:1. Daisy, to answer your question about why are biology mark schemes so specific, the mark schemes are specific because they were specific to the year that that exam was sat. Okay, the mark schemes change every year. The key marking points don't. So your key words, things like talking about whether it's homozygous recessive, heterozygous, whether it is a dihybrid cross, that doesn't change, but the mark scheme is specific to that year's exam and to that cohort that sat that exam at that time. So do make sure when you are looking at those mark schemes, you don't just memorize. That's where a lot of students fall down. They memorize the mark scheme. They then think that they can regurgitate that straight onto the paper in a completely different year's exam, and they don't get the marks. So just be aware, you look for the key themes, the points, the basic bits, which are the key definitions that are applied in any of the questions, but don't memorize the mark schemes. That's not a good way to do it. Use the mark scheme, absolutely. So the stuff that's in bold, yes, brackets, no. If it's in bold, they're like key terms they expect you to know. If it's in brackets, that means that it's implied. So if it's in a bracket, it means that the examiner is going to not have to find that, but that should be implied in the answer that you've given. So it's making sure that you know the difference between what's actually expected. So underlining, bold, absolutely, they are terms you need to include, but it doesn't necessarily mean it'll always be there. So just be careful on that question that you're answering. Right. Why does autosomal linkage affect dihybrid crosses. Right, I'm just about to go into this. I want to do one last thing and then I'll cover the autosomal linkage side of things. So if I go, so we've done a test cross, we've got yellow and wrinkled, green and round, and what we can see is these ones are different. So yellow wrinkled, capital Y, lowercase y, that's heterozygous, but that one obviously is homozygous recessive. Green and round, lowercase y's, so there's the two recessive, and then we've got the heterozygous. So what we can see is different ones to the parent genes. Okay, so let me do this one. If I have, so before I move on to autosomal linkage, if I have a, what did I have there? I had the yellow and round. So Y, Y, R, R, and I have, I've got to think this one through because my brain's not working as well today. And R, R, actually, I want to change that. If I have lowercase, capital R, lowercase r, capital Y, lowercase y, small r, if I were to rearrange this, capital Y, small y, capital R, R, and a Y, Y, R, R, what would I get? I would expect a 1 to 1 to 1 to 1 ratio. Students seem to think that if they have a individual like this, then I'm looking at it being a hetero versus a hetero. And therefore they expect a 9 to 3 to 3 to 1 ratio. That is not correct. It is not a heterozygous versus a heterozygous. They are heterozygous for that, heterozygous for that. They are homozygous and homozygous. So it's like you doing a heterozygous versus a homozygous, and therefore the expected ratio would be a 1 to 1 to 1 to 1 ratio, rather than if we had hetero with hetero, because hetero with hetero would be capital Y, lowercase y, capital R, lowercase r, and same again. So please be careful when you're reading what they've told you, because that's one of the high level questions that they will ask, where they're going to trip you up because you've read that there's a heterozygous there and a heterozygous there, because the heterozygous genes are in different individuals, but it acts like as if you were doing just one heterozygous versus a homozygous recessive. So just be careful on that one. Okay, Amna, you asked about autosomal linkage and how that affects a dihybrid cross. So I'm going to do this very basic with my drawing to start with, and then I'll bring in some of my other notes. So you know that you have got your chromosomes, which are linear. So if I have my genes here, and I have my same genes here. So we're going to stick with the yellow and the rounds, okay? So I'm going to use the red to show you that this is yellow and that this is round. And I'm going to show you that this is my, I've just said yellow. I didn't mean yellow, did I? I meant green. So I should be using G. Oh, no, that's because I was talking about the seeds. Yeah, I'll stick with the seeds and I'll stick with the yellow. So yeah, I'm going to go yellow because it's the dominant one. And then this one would be, the green would be recessive and the seed would be wrinkled. So I've kept the same allele combination that we had before. This time, however, they are joined on the same chromosome. So because they're joined on the same chromosome, what that therefore means is when I do my replication, so that's one chromosome. Oh, no, 1 second. Do that, do that. So now I have, through replication, I've now created my sister chromatids and my sister chromatids, which are held together by my centromere. Same again. There we go. With my very dodgy drawings. But what you can see here is they are now joined together. I've got my sister chromatids because DNA replication has occurred. So this was DNA replication that we did. We've taken the chromosomes, we have replicated them. We've replicated them by semi-conservative replication. And therefore, we now have identical sister chromatids. So if we have got the autosomal linkage, that means on chromosome, let's call this chromosome number one, because they are homologous chromosomes, they are going to have the same genes at the same location, so loci, and they're going to be the same length. So we know that we have got capital Y, the dominant allele, is going to be with the capital R, the round seeds. So what we then need to think about is, well, if that's the case, when they go through that meiotic division, remember, meiosis is about changing them from a diploid to a haploid. What I'm going to get is I'm going to get haploid cells that have half the genetic information. So therefore, that's what you can have. So if you have a dihybrid cross, and we're looking at it with regards to our capital Y, capital R, our lowercase y, lowercase r, lowercase y, lowercase r, that means that you will actually inherit those two together. So it works a bit like a monohybrid cross in that you can only either have the dominants together or you can have the lowercase together. And then people are like, well, how does that work? Because you get phenotypes of all the different individuals. So let me just, no, I didn't want to do that. I wanted this and this. There. So now if we go here, so here I have my individuals, but remember during prophase 1, these line up as bivalents. So what can happen is they can line up as bivalents and they can end up with crossing over occurring. So this is prophase 1. During prophase 1, crossing over can occur between non-sister chromatids. So what that basically means is they are going to exchange their genetic material. So what I need to do is rub these two out. And what could happen is I can end up with a capital R there. And I can end up with a lowercase r here instead. So homologous chromosomes are the same genes at the same locus, and they're the same length. They're still the same length, they're still the same genes, and they're still at the same position, locus. You're thinking about it, about the allele, different versions of the same gene. So whether it's a capital R or whether it's a lowercase r, that's to do with the actual DNA sequence So did the DNA sequence, let's say, go TTAA-TCGA-TGA, or did the DNA sequence go TTAA-CCG-TGA? Let's just say. I've just completely made that up. I have no idea what combination that would be. But what I'm showing you is that sequence is different. So because that sequence is different and it's only in one location that it's different, so we've got a point gene mutation because I've changed that position. I've got a different phenotype. I have a different version. I have the recessive allele rather than the dominant allele. And that's why mutations can actually cause variation. That makes sense. And the key thing, Amna, to understand on this one is because they don't follow Mendel's law, if we had, and we go back to our numbers, so what did I say, 1,400, if I were to use the same example, I would expect maybe about, I don't know, 680, maybe 700 here. No, that's completely wrong with my numbers. Try that again. I would expect 500, let's say 500 there, and I'd expect 560 here, let's say. So therefore, if I've got those two together, I now have 1060. So what have I left myself with? 340. 340 is 150 plus 20. 170, 170, let's say. So I would have a different number. We don't have a 1 to 1 to 1 to 1 ratio. We don't have a 3 to 1. What was the 3 to 1 to 1 ratio? Let's try that again. A 9 to 3 to 3 to 1 ratio. So we don't have the 9 to 3 to 3 to 1 ratio. We don't have the 1 to 1 to 1 ratio. So therefore, we can assume that they are autosomally linked. That's one assumption we could make because they don't follow Mendel's law of independent assortment. It could be that it's epigenetics that's involved, but we don't know because I've just made it up on the spot. So it's really important that you look at the question that they've given you and you use that to figure out what the expected ratios are going to be. How do you know which combination will be more? I just made it up. I don't know. It would just be a case of that they would have done it where they've actually bred them and got a number. I'm just making the numbers up on the spot. So if I give you an example, a specific example, which is to do with the linkage, so let me bring in a, here's one I prepared earlier for you. So let me bring this one in. So this is where they could use chi-squared side of things, where they take two heterozygous organisms, a purple flower with long pollen, that are going to be crossed. So we know that they're heterozygous. So because we know that they're heterozygous, because they told us it was two heterozygous individuals, we should know that our expected ratio, therefore, is a 9 to 3 to 3 to 1 ratio. But you can see here the numbers aren't following the 9 to 3 to 3 to 1 ratio. So already I can see something's not right. So then I would say, okay, the total adding them together is 6,952. I divide that by the 16, so 434, and then I work out my ratios, which are my expected. I then substitute that in. So it will be in the exam, if they give you an example, that will be, they've given you the numbers, and then you're trying to see if it matches with what you expect, the 9 to 3 to 3 to 1 ratio, or if it were a heterozygous versus a homorecessive, we would expect a 1 to 1 to 1 to 1 ratio. And then you'd compare those numbers together with that. And then obviously, You'd work out your chi-squared value, which is that. Now, they did do chi-squared in last year's paper. It doesn't mean they're not going to give another chi-squared test. However, it is one of those things to think about. If they've given you a chi-squared, they may not do it, but then they might be mean and go, well, they think we won't do it, so we'll do it again. So just make sure you do know how to do your statistical tests. Yes, it's still possible. It depends why you got the U grade in your mock paper. So what was it that resulted in you getting the U grade? Have you gone through the paper and seen what happened to why you got the I mean, that's a given for most students. I didn't revise, but it's not just about whether you revise, because there's a difference between have you been doing the revision or did you not cram for the exam? So be aware, revision and cramming are two different things. Cramming, you are basically trying to shovel as much information into your memory to then regurgitate it in a very short amount of time. That's not the same as revision, and you're never going to do well if that's all you've ever done. So did you cram before? When you say you didn't revise, have you not done anything of learning outside the class? That would be the other thing I'd say to you. You have got, what, 7, 8 weeks left until the actual, no, you're AS, so you're much less. When are you? You've now got 5 weeks? 5 weeks until the exam. Yeah, I know. I'm sorry to say that. 7 weeks, it is crazy. But 5 weeks, I think you are, yeah, cramming's not going to work. But if you're going to put the effort in now, there is still a possibility for you, especially for the AS level, but you need to work for it. You need to put the effort in if you want to do it, not just cram. Cramming doesn't get you where you need to be. That's why you will have missed, because you probably didn't know the detail you needed to go into to maximize your grade. So it's that whole thing of memorizing won't get you anywhere. Students think memorizing will get them lots of places. They won't. Maths practice, practical skills, and also application questions. And try and do as many past papers as you possibly can would be my suggestion. But it's still doable, but it's whether or not you want it and how much you are going to work for it. That's the key thing. Everything is possible when you put the effort in. For the next 5 weeks, basically your life is just studying and studying the right way, not the wrong way. Don't waste time on stuff you don't know. Guys, I'm probably going to do 5 more minutes. Is there anything else specifically you want to cover quickly before I close off for tonight? No, everybody is good for the evening. Key thing I would say is do make sure you are said they will make a past paper instead of AS exams. Who should I do? Ah, hold on. When you say your AS exam, are you talking about a mock exam that you're having done in the class in school? Not the actual AS exam. Right. Okay. Bacteria in the nitrogen cycle. Andreas, give me 1 second and I will do that one. Let me just answer this question. Do I think you should do the AS papers? No. Don't just memorize those papers. You will already have seen the questions potentially that they'll use, but what they will do is they could change them slightly. So if you try and memorize those mark schemes thinking that you're going to get a good grade at the end, that's not going to work. Use the time wisely to study the stuff you don't know. That will make a big difference for you. If you just try and kind of scam your way through, you're not going to do well. Any tips for writing questions? When you say writing questions, you mean writing the answers to the questions or actually writing questions? Rhizobium and Azotobacter, but I can't remember the nitro ones. So the Nitrosomonas I hate doing this because I'm so bad at spelling. Nitrosomonas. Somonas and Nitrobacter are the two that you need to know with regards to which one. So you obviously have your ammonium going to be converted into your nitrite, and then your nitrite, which is going to be converted into your nitrate. Yeah, so ammonium to nitrite and then the nitrite to nitrate. So with regards to that, that one is just a memorization. I don't think I've got a way that makes it easier to remember it because it's in ecosystems. I think it's just that I've memorized it in my head. Let me think. Hold on. Put me on the spot. I'll think about it. Let me think. So I've got to visualize and think about how I would do it. But when you're doing the nitrogen cycle and you've got the different parts, so nitrogen fixation, rhizobium is easy because of the roots, and azoto is in the soil. That they are the easy ones. But the nitrosomonas, let me think. How can you make it easier? Nitrosomonas, nitrite, NO2, NO2, Nitrobacter, NO3. See, I'm looking at it and I'm thinking, how can I use the alphabet to help me? Is there anything to do on that one? But it's not because this one has 3 O's, whereas that one goes up there, so that might confuse you. Nitrate. Ah, got it. There we go. Nitrate. Okay. See, this is the weird way that my brain works, trying to find something. S starts with nitrous ammonium. How does that help with the nitrate rate? R-A-T-E. I know it sounds ridiculous, but that's what I would do if I were trying to find a way to remember it. Nitrate is with the nitrobacter. Bacter has the A in it. Just someone says an A in it. Okay, that doesn't work. But it does spell rate. That's the only way I can think of it. There you go. That's my little way. I'm like, how can I do it? But yeah, I've just always memorized it. Nitrosomonas, nitrobacter, as a way of remembering it. But that will now be my new way of doing it. It is funny because I learn things all the time. What was it one of my students said to me today in the session? Oh, what did they say? And I was like, what do you mean by that? Posy. Is it posy? Posy pump. No. No. Nosy? Knocky pump? Is that what they were saying to me? See, I've even forgotten it now. Yeah, honestly, my brain has completely turned to mush. Um, you thought it was Super Nanny. You thought what was Super Nanny? Please don't tell me you thought I was Super Nanny. That's a bit of a worry. But oh, what was I saying? Posy was like positive out. That doesn't make sense because it was to do with the sodium out and potassium in. Sodium out. Soppy pump. That makes more sense. I was like, what was it? The soppy pump. And I was like, what's a soppy pump? And I was like, and that's how they remembered it. So sodium out, potassium in when you're looking at the sodium potassium pump. And I never thought about it that way. It's not stuck in my head. It probably isn't going to get stuck in my head because that doesn't work for me, but it worked for them. So the one thing I said to them is use it, but just don't write it. Make sure you don't write that and write it as being the sodium potassium pump. But yeah, it was like I was like, oh, was it? Um, so I'm actually going to, Alice, I'm just finishing up. The one thing I would say to you is if you are looking for epistasis, go check out my YouTube channel video. I've got a video on epistasis. Autosomal linkage would normally be with a chi-squared expected ratio, absolutely. Yeah, so dihybrids and monohybrids would be stats tests. Normally, if they're going to do those ones, it's normally to do with your chi-squared. I don't think they're going down the chi-squared test, but I could be wrong. Just to be clear, I could be wrong, but I don't feel they did a lot of chi-squared last year. I don't get to think they're going to bring in chi-squared again. I feel like there's definitely going to be statistical test in there, absolutely. I feel like Hardy-Weinberg's coming in more for genetics and patterns of inheritance and epistasis than I would say the chi-squared test. But like I said, I might get it right, I might get it wrong, I don't know. All I can do is I can do my research, which I've done, and tell you what I think, but that doesn't mean that I'm right. So please make sure you revise it all, make sure you're confident with it all. Yeah, I agree. I agree. Paper one was the hardest paper last year. Paper one, paper two were horrible. Paper three, would say would be easier. There'll always be one of the papers that's slightly easier. Which one? Nobody knows. So it just depends which one they decide to do. And that's just to do with the way that they've asked the questions, because they have to try and give you a broad array of questions in all the different papers. And because of that, if they've gone really hard on one question in one of them, they'll then go slightly easier on another because it has to be balanced. They can't just give you them all exactly the same. But I agree, paper one was quite hard. There was a lot of like, see, it wasn't actually that much application, but there's a lot more reading involved in the paper one last year than they have done in the past. And I think they're going to do that again. It's going to be comprehension. It used to be in the old style exams, it used to be that you were given like a paragraph that you had to read and then you had to answer questions related to that paragraph. So instead of them doing that, now what they seem to do is they seem to give you like a lot of information, a table or a graph of results, and then questions intermittent between. Yeah, AQA definitely still have the comprehension, but it's not as bad as what it used to be in the old style OCR and AQA. It's funny when you see it all come round in circles, the different exams that are done. Guys, thank you so much for joining me. I will be back on Monday again for the live session. And yeah, thank you. You too. Have a good evening, everybody.